Okay, so I decided let's just do this on the slide. Okay, so mechanisms of topoisomerase 1A and 1B. What we have here is that the topoisomerase has got a key tyrosine that's going to act as a nucleophile, and it's going to break phosphodiester bond that's in the DNA background. So it's the phosphorus is at phosphorus 5, got a lot of oxygens around it, so it's going to be an electrophilic phosphorus. And what happens is the nucleophile attacks the phosphorus, and part of the DNA strand leaves, and this is what is breaking the DNA strand. And so that one of the DNA strands is now broken, and the other part of it is now, the other half is now attached to the enzyme. When that happens, it can start to uncoil. The strand can start to uncoil because they've been broken. So that's and it's a phosphodiester, right? Because if this were a carbon here, this would be an ester group, and with two of them, it's diester. Okay, so the way that this can work is basically what the topoisomerase does is it attacks and it breaks one of the strands. The strands at least partially attach to the enzyme. It can move those strands around, basically, each other, and then it religates. It puts the cut that it makes back together. It fixes the cut. And so here we have mechanistically, it can both take two loops that are looped onto each other, and it can also just take a big coil of DNA and change the coiling. Okay, so there is two forms of DNA, there's three forms of DNA, but these are the main forms that we'll see and talk about. zDNA is something pretty exotic, basically. But we have these two different forms of DNA, and we have these glycosyl bonds in the DNA. And so again, this is the glycosyl bond. This is, again, I don't worry too much about zDNA. There's three classes of DNA interactive drugs. There's reversible binders, that form reversible interactions. There's alkylators that are going to react covalently with the DNA and alkylate them. And so we use these both as chemotherapeutics and also as mutagens. In which case they mutate the DNA because they create different shapes, basically. They alkylate the DNA so the DNA bases look like one DNA base looks like a different DNA base. And these shapes, the DNA bases are red based on their shape. There's also strand breaking DNA interactive drugs that break the DNA strands, and if you break the DNA strand into little pieces, the DNA can't replicate. So one of the things is that how do drugs interact with the DNA packed as chromatin? And so remember before that it was associated with those histone proteins. And so the nucleosomes, those little beads on the string of the chromatin, right, is DNA that's wrapped around the histone protein. But the reason that this happens is that the histone protein, so the histone forms a bihetero octamer, basically. And the histone proteins are on the inside, the DNA is on the outside quite beautifully. So that means the outer surface of the DNA is accessible, whatever strands face in the outside, the inside's facing the histone, but they're outside of so here's the DNA here in white, and all the beautiful colors are the histones protein that it's wrapped around. So it's pretty, so a drug can come in here, be like, sweet, got a major groove. Sweet, got a minor groove. Awesome. And the drug can come in there and bind. Also, like we talked about before, is that if you wanted to get a gene to express, right, no gene can express like this. It needs to create some space. It needs to create some space in between it and the histone, right? Because remember, you're going to have that DNA get turned into RNA. and the RNA is going to get turned into protein, right? So in order for this DNA to get turned into RNA, it has to be able to be pulled apart. It has to be able to be separated enough that you can have a DNA-dependent RNA polymerase that can use that DNA as a template, right? Here, this DNA ain't doing nothing but being coiled. So if you want the DNA to be able to show what bases are in it to an RNA polymerase protein that's going to polymerize RNA, you need to have it more accessible. You need to have some slack, basically, in your telephone cord. And so that's happening if a gene's getting expressed, there's a ton of slack, and that slack that's in the line makes it even easier for a drug to bind. There's a bunch of slack in this line, there's a bunch of kind of dynamics and things are moving around. It's even easier for a drug to kind of induce a fit, if you will, to find a place for itself in there. Okay, there's 3 ways that small molecules can reversibly bind to duplex DNA. So they can bind to the external electrostatics, and what we mean here is the phosphodiester backbone. They can bind into the groove, major or minor groove, right? They can sit in there, or they can intercalate. They can go in like a piece of paper shoving itself into a stack. They can fit in between the bases. So that's called intercalation. So external electrostatic binders, so these are cations. So right, so the phosphodiester backbone has a lot of minus charges, so you want a cation to associate with it. Groove binders are basically molecules, be they protein or small molecules, that are going to associate, just fit real nice into the grooves in the DNA base. And so an example of this is neotropsin. And so this is a drug that's going to fit in with a minor groove. DNA intercalators, so this is a broad class. They're generally flat and aromatic or heteroaromatic. They don't do anything for the H bonding, but they do, they stack in between the base pairs and they mess up the helix and they help unwind the DNA. And so they mess up topoisomerases, they mess up DNA polymerases, and so when they mess up the topoisomerases, that means the DNA isn't properly wound, and this can lead to apoptosis, among other things. They mess up the DNA polymerase, so this is how the cells replicate, and this is how also the DNA chain is elongated, so more DNA basically can't get made, so that can lead to apoptosis or just eventual cell death as well. So basically the way this works is here that it sticks in between the base pairs, and it forms, these are non-covalent interactions, but it messes with the overall topology of the DNA. So there's also, so for example, a lot of potent chemotherapeutics interact with the DNA topoisomerase 1 complex. An example of this is topotecan, and topotecan associates with DNA when it's bound to topoisomerase 1. So this whole step, if we go back here, this whole process takes kind of a long time. You can imagine, right, what you're doing, because remember, again, this is an enzyme that's doing this to 1.84 meters of DNA, right? And it's an enzyme. So this kind of takes some time. It takes some ATP, it takes some effort. And so while this is going on, something like the topotecan can come in here and it can associate with this complex. It can associate with how, because remember that the DNA, right, that the topoisomerase 2 has formed a DNA protein bond through part of this mechanism, right? That's formed a bond here through this part of the mechanism, like when we talked about, I have lost my place, talked about here, right? So it's formed a covalent bond. So something like topotecan can get in here and associate with this in such a way that it's going to end up inhibiting the enzyme. Okay, so that's for DNA intercalators. DNA damage is not again, it's not it's not enough to just intercalate. You need to mess up something that by act of being there associated with the DNA, you mess up something else. That's what an intercalator, it needs to be a good enough DNA intercalator that by being there, it's strong enough bond that it's messed up either the topoisomerase or the DNA dependent DNA polymerase, DNA polymerase 2. And so here's some examples. These are acridines. These are dyes. Actinomycins. So these come from soil bacteria, and anthracycline. And so this is these are very common ones because doxorubicin, for example, is a component of a lot of leukemia and other combination chemotherapies for a lot of different drugs, or for a lot of different cancers. And daunorubicin is as well. You can imagine they both have the word Latin word rubris in them, and they're red. So certainly acridine, some of these are also antibacterial for the same reason. But using this antibacterial, this is an example of a lead modification, and they're able to come up with an anti-leukemia, so anti-human cell, essentially, drug that stabilizes the topoisomerase complex. Dactinomycin, this is another an actinomycin tumor, anti-tumor. And so what we have here is we have basically the peptide and the chromophore. What do you mean? So you're saying, what do you mean by peptide and chromophore? So the way that this is kind of hard to maybe understand the picture if you're not familiar with it. Okay, so right here is a phosphate. Right here is a phosphate. This is a sugar. That's a sugar right there. That's a sugar right there. That's a sugar right there. This is edge on. This is a DNA base. This is another DNA base that's H bonded to it. This is a DNA base. So this is your A or T, and this is your A or T, and this is your G or C over here. This is your G or C over here. And so what happens is that if we look back to these actinomycins, they have all these actinomycins have peptides hooked up to a heteroaromatic compound. And this heteroaromatic compound, we call this chromophore in this case because it's colored. So this is going to be a light-absorbing, colorful compound, red or yellow or something like that. But this has got the necessary conjugation to interact with visible light. And then it's got these peptides here. This is the portion that's going to intercalate with DNA. And so we can see this here in this crystallized complex. So this is DNA going one way, this is DNA going the other way. But it's a DNA, it's duplex DNA. And then we have here, this is an edge-on view of the chromophore. So it's an edge-on view of that heteroaromatic complex. And then going into the back of the screen behind your monitor are the peptides associated to the chromophore. So this is turned 90 degrees so that essentially these methyl groups in this case would be pointing towards us, and these guys would be pointing back out of your monitor. And so here you can imagine that's really flat, and it's basically the width of DNA bases and their associated H bond approximately. So it fits really nicely in between two H-bonded DNA bases. And then it's got all these peptides, it's got all the spinach that hangs out on the outside, and it can mess with DNA replication and topoisomerase 2. Which is, we haven't talked about how topoisomerase 2 works, but topoisomerase 2 is the one that can break both DNA strands. So we just talked about topoisomerase 1 that breaks one DNA strand, topo 2 breaks both. And so we can stabilize. So here we have it flipped, right? So here's a peptide coming out. One of the pin-type, sorry, this isn't a pentapeptide. Sorry, this is a sugar off of a doxorubicin. So I'm sorry about that. This is actinomycin, but this is that common theory. This is basically an anthracycline. So we call these anthracyclines. But what that picture is showing you is that this guy is flipped on the side. That sugar group is coming out at us. This part of the molecule is coming out at us, and this part's going back into the screen. So this is for doxorubicin. And so if we look here, see, this is that sugar group, and that ring that was unsaturated is coming out at us, and then the rest of the rings are intercalated into the DNA grooves. And so even the that carbonyl on those anthracyclines can now essentially H bond with portions of DNA bases. So this is how this creates it. Basically, it's stabilized through both pi stacking energy and H bonding energy. There is ways that molecules can intercalate into two DNA strands, but this is a little more esoteric and not common. Okay, so one of the original DNA drugs, like we've talked about before, is nitrogen mustards. So these are still used in some way or another today, which is one of the reasons we need to all invent new drugs, right? Is that and the way that they found it is that this is sulfur mustard. This is mustard, one of the gases that we would call mustard gas when we're talking about World War I. They also, I think, used it some in World War II, but anyways, this is in World War I. And one of the things they found was that they see leukopenia. So they're seeing that the bone marrow is getting destroyed, and they see ulceration of GI tract, which is another thing that we see actually with a lot of different chemotherapeutics, is this kind of GI tract issues, and those are rapidly replicating cells. And so this is lead discovery. So they have their lead, and it's by basically just fortuitous observation. And they began to modify this. So this is the beginning of cancer chemotherapy. They use this for Hodgkin's disease, so that's a lymphoma. So it's a cancer, Hodgkin's lymphoma. So basically, it's a cancer of cells in your bone marrow that make white blood cells. And it's also, so it's related to, it can also be a cancer of the lymphatic system. So basically still related to moving white blood cells around your body. There's nodes called lymph nodes that are going to be moving a lot of those immune cells in your body in a similar way to veins, but there isn't the same circulatory pressure, right? There isn't the same pumping action, but you have ways to move white blood cells around. In any case, you can imagine that they're like, okay, if people have an overproliferation of white blood cells, we should use this drug that we know causes people to lose a lot of white blood cells, right? That makes sense. Let's use that as a anti-cancer drug for people that have cancers with essentially too many aberrant white blood cells. So the chemistry of this we've actually talked about before, which is that we're going to do, an alkylation reaction. And here we will do a breakout slide for real.